Either you or the person sitting next to you has probably used some form of contraception. But think about how many choices women have when it comes to avoiding pregnancy, while men have only two. On the face of it, regulating fertility in men should be the more obvious choice, since sperm is constantly produced and not in cycles like eggs. According to the National Center for Disease Control Prevention, 48% of pregnancies in America are unwanted, half of them which are handled through abortion. Imagine a molecular contraceptive effective for both men and women with no hormonal side effects. We've identified two proteins that can pave way for this possibility. His35 and HTOS1 are histone proteins that wrap our DNA and behave as bodyguards to genes. They generally control whether transcriptional machinery can access the genes and thus turn them on. So, how do we know that these histones can be targeted and utilized for universal contraception? For starters, His35 is found in both sperm and eggs, while HTOS1 is found only in sperm. It's interesting. When we inhibit HTOS1, fertility declines by 17%. And when we inhibit His35, fertility declines by 30%. But what happens when we inhibit both histone proteins simultaneously? Unexpectedly, fertility is 100% suppressed in our model organism, C. elegans. So the question of how do we develop a molecular contraceptive more closely becomes how do we work backwards from these results, harness the activities of these histones, and develop a fertility switch that could be utilized in humans. The purpose of my research is just that. What gives me hope is currently human relatives of these histones are serving as targets for breast cancer therapies. Now, during my time here, I aim to really understand what these histones could be possibly doing and learn why they are essential for fertility. I'm doing so by 1, identifying their contributions to genes that establish the sperm and egg identity. Are they turning them on or off? And second, I'm characterizing dynamic mechanisms during fertilization. Where are these proteins going and how are they moving during this time? Answering these questions will pave way for molecular contraception. And for all the men and women in this room, be prepared to share this responsibility. Thank you.